Okay, so hi everyone. My name is Devin Weir, and I'm Vice President of the AMS and a research scientist and taxonomist at Manaki Whenua Lanke Research in Auckland, New Zealand. I'd just like to welcome everybody here for our June virtual seminar. Just gave it a bit of a plug about our joint AMS NZMS conference, and there's a slide up there at the moment about that. And I'll post a link to the website in the chat later on as well. But yes, be in November in Wellington this year. But on to today's seminar. So our speaker today is Camille Truong, and she is a mycologist at Royal Botanic Gardens in Victoria. She has a PhD from the University of Geneva in Switzerland, and she was a postdoc both at Duke University and at the University of Florida in the US, and a researcher at the National Autonomous University of Mexico. She's conducted research in the temperate and tropical forests of the Americas, Africa, Australia, and her expertise spans across the taxonomy and molecular systematics and genomics and microbiology. And she's particularly interested in the evolution of plant and fungal interactions throughout time and space, and the role of fungal diversity for understanding ecosystem function. So today we'll move on to the talk and just the usual kind of thing. So just ask any questions through the Q&A or chat if you like. And at the end of the talk, there'll be a brief survey. But yeah, up to you. Please take it away, Camille.
Okay, thank you very much for the nice introduction. Hello, everyone. And so before I start, I would like to acknowledge the traditional owners and custodians throughout this land and pay my respect to elders past, present, and emerging. And I also recognize the cultural and scientific knowledge of Australian fungi held by Aboriginal and Torres Strait Islander peoples for millennia. So today I'm going to tell you about a project that I started some years ago already, looking at the role of ectomycorrhizal fungi for nutrient cycling, and in particular the nitrogen economy of Nothofagus. So I recently arrived in Australia to take up a position at the Royal Botanic Gardens Victoria. But for this project, I started as a postdoc at the University of Florida, and I'm still collaborating with them about this. So when we think about the role of ectomycorrhizal fungi for nutrient cycling, we have to look a little bit deeper into plant nutrition. So of course, we all know that the plants produces sugar and so has a source of carbon from photosynthesis, but the plant also need water and minerals. And so minerals such as nitrogen, but also phosphorus or potassium, for example, the plant can only absorb it in an inorganic form. So if you have nitrogen or phosphorus that is trapped inside organic matter, so for example, in leaf litter, the plant doesn't have access to these sources of nitrogen or phosphorus. And so in order for the plants to have these inorganic minerals, it needs the mineralization of the organic matter by microorganisms. And this is done mostly by fungi, but also by bacteria. And then for mining, so for searching in the soil for these minerals and then transporting to the plant, this is enhanced by mycorrhizal fungi. So basically, as you obviously know, the plant is trading carbon, so it gives carbon to the mycorrhizal fungi in exchange for these minerals, but also sometimes water. And the mycorrhizal fungi can provide other services such as resistance to certain stress or pathogens. And so all the mycorrhizal fungi, they trade this carbon against the nutrient, but they do it sometimes in a very different ways. So for example, the arbuscular mycorrhizal fungi, they can give this mineral to the plants, but they cannot mineralize the organic matter. They stimulate the mineralization in the soil by secreting sugars so that other microorganisms are going to mineralize the organic matter, and then they transport it to the plant, but they cannot do the mineralization themselves. On the other hand, some group of ectomycorrhizal fungi, so not all ectomycorrhizal fungi, some of them, especially the genus Cortinarius, they can degrade the organic matter to access organic forms of nitrogen. And we know this based on genomic studies that studies enzymatic pathway, and we know that these ectomycorrhizal fungi, they have enzymes to degrade organic matter. And so even though, because they can degrade organic matter, there have been a little bit of confusion in recent years, saying that, oh, so these ectomycorrhizal fungi, maybe they have saprotrophic abilities, but it's really not the case. And because both these ectomycorrhizal fungi and the saprotrophs, they degrade organic matter, but the saprotrophs, they do it in order to get a source of carbon, while the mycorrhizal fungi, they still receive the carbon from the plant. So they do degrade organic matter, but not to get carbon. They do it to get nitrogen. So they're not saprotrophs. And another difference with saprotrophs is that they also do it in slightly different ways. So in order to degrade organic matters, saprotrophic fungi, they mostly use hydrolytic enzymes, while mycorrhizal fungi, ectomycorrhizal fungi, they mostly do it through oxidation. So it's also a different way of degrading the organic matter. And so what is a nutrient economy or nitrogen economy? So there's been a lot of literature recently, also for the public, about mycorrhizal fungi, which I think is a great thing. But on the other hand, there is a little bit of like this romanticism about the relation that plants and mycorrhizal fungi have, you know, like this great connection. And it's a lot of people compare it to like a love affair. As scientists, we know that it's not really, it doesn't really have a lot to do with love, but it's more a business thing. So basically what happened is like, it's really a trading thing. So the plant is going to trade carbons for this nutrient. And so it is known that the more the mycorrhizal fungi give, the more molecules of nutrient the mycorrhizal fungi is going to give, the more the plant is going to give them. So it's really, that's why we call it a nutrient economy, because it's really like a market. And there have been, in recent years, there have been studies that took like economic models to try to apply this to mycorrhizal fungi, and it actually works. It's really interesting. So basically, this trading system, studying the nutrient economy, is studying this trading system between a plant species and the community of mycorrhizal fungi that are in the soil. And so, of course, this trading system is going to depend on the soil fungal communities that are present in the soil. Competitors, of course, they compete between each other. They compete with saprotroph fungi as well. And under certain condition, mycorrhizal fungi maybe is going to be better at getting this nutrient for the plant. So under certain condition, he will receive more carbon. And then if the condition change, then another species may receive more carbon because it's more adapted. So that's how the tree is actually able to adapt to environmental change. And then all these things, of course, it depends also of the nutrient availability to the plant, because if there is a lot of inorganic nitrogen that is readily available to the plant, the plant doesn't really need the mycorrhizal fungi anymore. And so all these also depend on what are the general environmental factors that we find in the soil. And so this is really interesting because, like I said, this is not really a love affair. It has been shown that, for example, if you give fertilizer to a mycorrhizal plant, and so the plant doesn't really need the mycorrhizal fungi to get nutrients, it actually gets rid of them. And so this ecological market, let's say, on the ectomycorrhizal symbiosis, it has been studied quite extensively in recent years, but mostly in the northern hemisphere and with a model that was mostly coniferous forests, so pines and spruce. And so what they showed is that they tried to explain why in a lot of temperate forests, the nitrogen in general is highly limited, even though we have these large trees everywhere that have obviously high demand in nitrogen. Right? And what they show is that when you have high soil nitrogen availability, the plant actually gives less carbon to the mycorrhizal fungi in exchange for the nitrogen. But when there is low soil nitrogen availability, the plant is going to give more carbon, allocate more carbon to the mycorrhizal fungi, even though they don't necessarily give more, they don't necessarily transfer more nitrogen to the tree, but because the plant is craving for nitrogen so much, it's trying to kind of enhance it. It's like bargaining, kind of, if you want to say. And so basically what has happened is that you have if you have a reduced nitrogen availability, so very few nitrogen available for the plant in the soil, then the plant is going to increase the carbon allocation to the ectomycorrhizal fungi. And because I was telling you of this group of ectomycorrhizal fungi, especially Cortinarius, has this ability to bypass the mineralization. It can degrade, take the nitrogen from the organic matter and give it to the plant directly without doing mineralization. So it's actually increased the immobilization of nitrogen and increased the reduction of the soil nitrogen availability. And that's why they say the forests are actually trapped in this nitrogen limitation, because there's very little nitrogen available in the soil. And so the plants rely more and more of these ectomycorrhizal fungi that actually increase the nitrogen limitation. And so interestingly, it has been shown in recent years that this is also probably what is favoring the dominance of these ectomycorrhizal species in the forest. I mean, we see here when we have eucalyptus forest, for example, it's really these ectomycorrhizal plants, they really dominate in this forest. The same happen in the northern hemisphere with like oak forest or pine forest. And so we think that this reduction of the nitrogen availability, it actually impairs the colonization of plants that don't associate with this fungi, because as I was telling you, the arbuscular mycorrhizal fungi, they are not able to degrade organic matter, so they need mineralization. And so if there is no mineralization, the arbuscular mycorrhizal plant basically cannot survive there. And so this study showed that if you actually increase the nitrogen available in the soil in response to anthropogenic impacts. This is happening a lot, for example, in Europe and in the US because of air contamination. They see that because there is more nitrogen available, there is this shift of the ECM dominant forest towards the arbuscular mycorrhizal forest. So the forest is actually changing in response to this increase in nitrogen in the soil. So this is to give you a few examples of how important and how connected there is the relationship between the fungi in the soil and the vegetation that we see in relation to this balance between the nutrients that are available in the soil, but also with the different strategy that mycorrhizal fungi have developed to give it to the tree. And so all of these models, they have been mostly studied in the temperate and boreal zone of the northern hemisphere, and they have been particularly studied in coniferous forests. And so in this project, we were interested to see what is happening in the southern hemisphere, where the ectomycorrhizal forests are dominated by angiosperms, mostly Myrtaceae, like Leptospermum, eucalyptus, and in Patagonia, mostly the family Nothofagaceae. And so we were interested to see what is the role of these ECM fungi for the nitrogen economy of Nothofagus in Patagonia, and to see if we can find similar patterns that have been shown in the northern hemisphere. So for example, we wanted to test if the nitrogen availability is going to correlate with the nutrient demand of different Nothofagus species. And also if, as it was shown in boreal forest, the dependence on the ECM symbiosis will increase with nitrogen limitation. And so to give you a broad introduction about this forest, I'm going to emphasize a little bit on the difference with Nothofagus forest that we found in Australasia. So there, the native tree species, of course, are broad-leaved Nothofagaceae. And one major difference is that they're really the dominant forest tree there. So there's no Myrtaceae, there's no other ECM host occurring in this forest. Only ectomycorrhizal association are with Nothofagaceae, and they're really dominant. There are some conifers, such as the genus Podocarpus, Fitzroya, or Pilgerodendron, that all have a distant relative in Australasia. But they're not really dominant. They're usually emergent tree, and they also associate with arbuscular mycorrhizal fungi. So it's a different type of association. And another major difference that I see is that Several of the Nothofagaceae species in the subgenus Nothofagus that are dominant in Patagonia are deciduous. And so you see this really beautiful landscape in the fall that you would never see in Australasia. But I was lucky to go to Tasmania in May and visit some of the population of the Nothofagus guinea, what they call fagus here. So it was beautiful colors, but it's really patchy. So you really don't see a landscape like the one on the photo. And so, as you also know, there's Nothofagaceae species. They are part of what we call the Gondwanan flora or the Antarctic flora, because even though the present distribution is very fragmented in southern South America, Australia, New Zealand, Papua New Guinea, and New Caledonia, we know that when in past geological years, about maybe 100 million years ago, when Antarctica, Australia, New Zealand, it's not on the map, I apologize, and South America were still connected, we know that Nothofagaceae forests were occurring along the whole range and were actually connected to each other. So we find this Gondwanan connection or Gondwanan origin in the Ectomycorrhizal fungi associated with Nothofagus as well. This is one example, Ostracoxylistatum. It's a genus that is exclusively found in the southern hemisphere. Another example, for example, in the Ascomycetes is Germorium. The genus Germorium, it was recently synonymized, maybe you know it under the name Underwoodia. So this is another example of this southern hemisphere fungi. But when we did an inventory and barcoding of Patagonian fungi, and really in Patagonia, the most diverse and also the most abundant group in terms of fruiting body, but also in the soil and in the ectomycorrhizal root tips is really the genus Cortinarius. You see like more than 1/3 of all the collection we made were from this genus. And so it's huge diversity. It's very beautiful species. This is Cortinarius magnificus, which is, you can see it has this amazing yellow trama. Another, some small one, Cortinarius obtusii group. There are some species complex. So for example, Cortinarius magellanicus, it's really common species there. And we found based on phylogenetic analysis that it's probably several species that occur there. There are some species also that are hypogynous. This one, Cortinarius, I should say Taxterogaster magellanicus. This is actually the names has recently been reinstalled. And so it's actually a secordioid species. It has a little side, but the fruiting body is enclosed. So this is truffle. And so there's really a lot of diversity. There's hundreds of species of Cortinarius. I think we have about like 450 already that are listed. And so the ongoing work is done by Tuula Niskanen, which is our collaborator at the Kew Botanical Gardens. And I think when she came to Patagonia, she was really amazed by the diversity that we found. And another group that is interesting in this forest is actually in the Ascomycetes, in the order Pedzizales. We find a very interesting diversity of ectomycorrhizal lineages within this group. So usually people don't think of Ascomycetes necessarily as being ectomycorrhizal, but there are actually numerous lineages that do this. Example of cap fungi such as Alohina, Pulvinula, Amylascus, which is a truffle. And as you can see, Amylascus is the truffle, and it also forms these mitotic spore mats. So it's like a vegetative way of propagation. And so these Ascomycetes, they're very intriguing because they tend to heavily colonize seedlings. So we also think that they have a lot of role for forest recruitment. What is happening here? Okay, and so going back to the ecology of ECM fungi, we settled our plot in the archipelago of Tierra del Fuego. So we are here at the southern tip of South America, and we chose this because, as I was telling you, there's limited knowledge about ECM association in this group. Nothofagus is the only ECM host plant. So if we find an ectomycorrhizal fungi, we know that it is associated with Nothofagus because there's any other host there. We also choose to do our study plot there because the archipelago has very strong climatic and edaphic gradient on very short distances. Because as you can see, this is the tip of the Andes Cordillera here. So the southern part of the island, the archipelago, is very wet. You get 2,000 to 4,000 millimeters of rain or snow every year, while the northern side of the archipelago in the rain shadow, we see very little rain, and it actually there's not even forest anymore on the northern side. It's all steppe area, and so there are 3 Nothofagus species that grow there according to climate, water regime, and soil properties. So Nothofagus betuloides is the species that grow along the coast, Nothofagus pumilo in the interior along the mountain range, and then Nothofagus antica in the ecotone with the steppe. And so they really form monospecific forests of a single ECM host species. They are kind of mixed, I would say mixed forests. There is a transition zone, as you can see, where the species enter in contact. It's not really real mixed forest. It's more like a transition between two. And so the first species, Nothofagus betuloides, as I was telling you, is growing in oceanic area along lakes and major lakes on the coast. It's also an evergreen species. And so the soil is very acidic, it's seasonally waterlogged, and the organic horizon is sometimes really thin. As you can see, sometimes the trees, they almost grow directly on bedrocks. And so we also know from previous study that this tree has poor nutrient content of leaf. And so because of that, it was always assumed that it grows on nutrient poor soils, but it had never been studied before. And so Nothofagus pumilo, the second species that grow along the mountain slope, this time, this is a deciduous species. That's the species that I've shown you in the photos before. It's a widespread species in Patagonia, and it grows on soils that are deeper and also more well-drained. And it also has a higher nutrient content of leaves. So it was always assumed that it was growing on more fertile soil, but never tested again. And so this is here, you see what I was telling you about the mixed forest between pumilo and betuloides. And so it's really actually easy to spot them in the fall because you see that because it's deciduous, all the orangish, reddish color is Nothofagus pumilo, and the green is Nothofagus betuloides. So you see that they kind of enter in contact, they mix a little bit, but it's not really like a mixed forest in the sense that we would see maybe in the northern hemisphere. And then the third species, Nothofagus antarctica, is a species that grows in extreme habitat. So it forms the tree line, it grows in the ecotone with steppe or along bogs. It's also deciduous, and it grows on more basic and deeper soil. And as you can see, it's like kind of open forest. And so it's naturally grazed by guanacos, also by cattle. I mean, when there is cattle, but naturally it's grazed. And so it has kind of an intermediate nutrient content of leaf compared to the other 2 species. And so the assumption is that it's highly adapted to environmental stress. So that's why it's the species that you only find in like this extreme habitat, not because it cannot grow in other places, but mostly because in other species, the competition with the other 3 species is too high. And so there is also a contact zone between pumilo and Antarctica, not Antarctica and betuloides. They never go close by. But as you can see, this is not really a mixed forest because you have, so in the lowland along the bogs, you have Nothofagus antarctica. And then here when the elevation is slightly higher and then along the slope of the mountains, you have Nothofagus pumilo. So the hypothesis that we have is that we think that the fungal community structure in the soil and the functions, they are going to be mostly driven by environmental factors because based on preliminary results and previous study that we have done, these forests, they are heavily structured by the soil properties and the climate. And then also we want to test this hypothesis that I was telling you at the beginning, that the nitrogen availability is going to correlate with the nutrient demand of the different Nothofagus species. So there's more nitrogen available when the trees have more nitrogen demand. And then finally, we want to see if there is more nitrogen limitation, if we are going to find more dependence on the ectomycorrhizal symbiosis, same as what has been observed in boreal forest. And so in order to do this, we sampled, so there's 5 types of forests, so the 3 monodominant forests and the 2 mixed forests. We had about 3 population, 10 sample per population, so 150 sample in total, soil sample. And for each soil sample, what we did is identify fungal communities using the ITS1 barcode on the Illumina MySeq platform, so using meta-barcoding. We also measured edaphic variable for each soil sample, so the pH, the soil moisture, the carbon nitrogen ratio, and then the available nitrogen and phosphorus, so inorganic nitrogen and phosphorus. And then for the, not the mixed forest, but for the monospecific forest, we also measure the net nitrogen mineralization after incubation during 30 days, and the microbial biomass of nitrogen. So by microbial biomass, I mean the nitrogen that is not available in the soil, but present inside the body of the microorganism. You basically do a lysis of the microorganism in the soil, and then you measure the nitrogen. And then finally, we also measured the enzymatic activity of 8 extracellular fungal enzymes using fluorometric assays. And so in terms of fungal diversity, the first thing that we observed is that in the Nothofagus Antarctica forest, non-ECM fungi, so mostly saprophytic fungi, actually dominate in the soil, which is not really surprising because as I was telling you, the soil is deeper, there's grazing, so there's all these grasses, there's more saprophytic fungi there. While in the other type of forest, and especially in the mixed pumilo-betuloid forest, the ectomycorrhizal fungi usually dominate. And so as you can see here, this is the number of ectomycorrhizal O2 that we find. We see that, so the mixed forests, they actually have more diversity than the monospecific forest. We find patterns of differences for 2 families in particular. The family Cortinariaceae has like this, was quite abundant in all the type of forests, but particularly abundant in the mixed pumilo-betuloides forest, while Nothofagus Antarctica had the lowest abundance. And also we find a very interesting pattern for the family Clavulinaceae, which is another ECM group, where in the betuloides and the mixed forest, these fungi were actually very abundant in the soil. And it was quite surprising because it's not a fungi that we find abundantly fruiting, so we don't have a lot of fruit body collections from this, but they were very, very abundant in the soil on these 2 forests that are also the most acidic soils. And so in general, what we found is that the diversity of the ECM fungi, they increase in mixed forests, which makes a lot of sense because usually when you have an increase in the tree diversity, it means you're going to have more heterogeneous habitats, more heterogeneous litter, and this kind of favor the diversity of the ECM fungi. Now, if we look at the fungal community structure of ECM community and non-ECM community. So when I say non-ECM community, it means mostly, it's actually mostly 90% saprotroph. We don't call it saprotrophic communities because some of these fungi, it's sometimes not clear if they're really saprotroph. Some of them have been described as underphyte, so we prefer to say non-ECM. And so we see that for both of them, the pH was actually the best environmental factor to differentiate between the communities in the different forests. But not only, so there are differences in between the forest type that were not explained by pH. And so in mixed forests, we didn't find significant pairwise differences between betuloides, so and the mixed forest, and between Antarctica and the mixed Antarctica forest. So basically what is happening here in terms of community composition is that we have the 3 forests, Antarctica, betuloides, pumilo, but then in a mixed forest, the community looked like betuloides and looked like Antarctica, while the community that is associated with Nothofagus pumilo kind of in the presence of the two other tree species, the community structure actually changes. And we will see why, because the nitrogen economy of this forest is very particular. And so in terms of enzyme activity, what we find here in this case is that soil moisture is very important to structure the enzymatic activity in between the different forests. But also, again, there is differences between forest types that could not be explained by the factors that we used in the model. And so again, we didn't find significant pairwise differences between Betuloides with Pumilobettuloides and between Antarctica with Pumilo-Antarctica. So basically the mixed forests, they function in the same way of the monospecific Betuloides or Antarctica forest. And another thing that is important is between the tree species, we didn't find significant pairwise differences in mixed forests. And that was the same, so here I forgot to mention. So we tried to find if these differences that we see between the forest type are related to really like a specificity of this Eastern fungi for different tree species. But we found that in mixed forests, there were no difference between samples that were sampled at the bottom of a certain tree species or the other tree species. Of course, this is very preliminary, because if you really want to study specificity or preference, you would have to study roots and see really what are the fungi that are colonizing this wood and not the fungi that are in the soil, and we didn't do this. But the data that we have at hand don't really give us the impression that there is a specificity in between the tree species. Okay. And so basically the factors that are affecting fungal function, in general in this forest, we see is mostly pH and soil moistures. And so the pH is driving a lot of change in edaphic condition. So we see the pH, it correlates actually with all the different edaphic variables that we have identified. So it correlates with the availability of nitrogen and phosphorus, for example, while soil moisture it correlates heavily with the activity of the enzyme. When there's more soil moistures, there's more enzyme activities, which also makes a lot of sense. But as we can say, if we look at the variation partitioning of the enzyme activity between the different forest types, we see that, so even though there is some of the variation in the enzymes that is explained by the soil variables, there is still this variation between forest type that we don't know where it comes from. And it doesn't come from differences in fungal communities, because we didn't really find correlation between the two. So we think that there are other factors that probably take place, and most probably climate, because of this very different environment where the three monospecific forests occur. Of course, the temperature and also the precipitation probably play a very major role. And so now if we look a little bit more in detail in the nitrogen cycling, we find very surprising results at first. So as I was telling you at the beginning, Nothofagus pumilo have a high nutrient content of the leaf, while Nothofagus betuloides have a low nutrient content of the leaf. But despite this, we found that in Nothofagus betuloides, the mineralization of the organic matter and also the nitrogen available in the soil was actually much higher than in Nothofagus pumilo, even though Nothofagus pumilo needs more nitrogen, right? So the question is, so if Nothofagus pumilo needs more nitrogen and there's no nitrogen available in the soil, where does the nitrogen came from? And this is when it's interesting to look at the microbial biomass of nitrogens, because here we see that actually most of the nitrogen that is present in the soil was present in Nothofagus pumilo forest in the microbial biomass. So most of the nitrogen is actually trapped inside the body of the microorganism, in the mycelium, for example. And for Nothofagus Antarctica, we see this is a little bit more like an intermediate. It has medium nitrogen and it's like an intermediate thing. But really we have these really huge differences with Nothofagus betuloides that has very high mineralization, and Nothofagus pumilo that has very high nitrogen microbial biomass. And this pattern, it was interesting because it's also correlated with the richness and the abundance of one fungal ECM fungal family, Clavulinaceae, which is actually adapted to inorganic nitrogen uptake. So basically, when we have more mineralization, we have more nitrogen, and then when we have more microbial biomass, we have also less diversity of Clavulinaceae. And so this also correlated with the different forest type. And so remember, this family Clavulinaceae, it was highly abundant in the Betuloides forest, and that's why we can really see clearly here that it probably has a role in Betuloides forest for this very high mineralization that we see. And so in conclusion, what we can see is that Nothofagus betuloides is showing a clear pattern of what we call an inorganic nitrogen economy. So basically, we have low nitrogen content of the leaves, but relatively high mineralization and high available nitrogen. This is here in yellow in the soil, and also very low pH. And so what we have here is in a situation where we have this accelerated nitrogen cycling, where there is very low nitrogen. So the litter doesn't have a lot of nitrogen available, but it's very rapidly recycled by these fungi, but also within anoxic bacteria, because if you remember at the beginning, I was telling you that these forests, they can be temporarily waterlogged. And so probably also to prevent leaching, the nitrogen is very rapidly mineralized and recycled and back into the trees. And so in the case of Nothofagus pumilo, we have exactly the opposite. This is typically what is called an organic nitrogen economy. So where we have a high nutrient content. So basically the tree needs a lot of nitrogen going back to it, and then the litter has high nitrogen content. But we have this very, very low nitrogen mineralization in the soil, and also available nitrogen in the soil. But on the other hand, we have this really high microbials of nitrogen. And also we found that some of the enzymes that we have been studied, this one, leucine aminopeptidase, it's an enzyme that is specific for nitrogen release. It was also higher. So basically here, what we have is that this typical nitrogen economy that I was showing you in the boreal forest, where ECM fungi, and we tried to find pattern with Cortinarius, because Cortinarius in boreal forest has been shown to do this kind of thing. The thing is that because Cortinarius in Patagonia is present everywhere in all types of forests, we could not really find a specific pattern. Maybe we will have to look at some group of Cortinarius in particular. But we really showed that this organic nitrogen economy helped Nothofagus pumilo to have access to all this nitrogen, even though there is very low nitrogen available in the soil. And so that was really interesting, because this pattern is actually very, very similar to what has been shown in boreal forest, but it has been shown on conifer. And here we are with an angiosperm in the southern hemisphere. But the patterns and the type of fungi are actually very similar. And so finally, for Nothofagus antarctica, in this case, it's a little bit more intermediate. So we have, as you can see, you have some mineralization happening, you have some microbial biomass as well. And so remember that this is also a forest that has a higher diversity of non-ECM fungi. So we think that here the nitrogen economy is definitely not dependent on the ECM fungi. And so it's a little bit a different pattern. And so in conclusion, we did find that environmental factors are the best predictor for fungal community and function, but it's the one that we studied in the soil are probably not the only one. So we think that climate probably play a role. And then secondly, we didn't find at all that the nitrogen availability of in the soil correlate with the nutrient demand of the different Nothofagus species. It was actually the opposite, because Nothofagus pumilo, which needs more nitrogen, was actually the one that has the lowest nitrogen availability. But we did find that the dependence on the ECM symbiosis increased with the nitrogen limitation. And we are now conducting a study to see if this is linked to forest monodominance. So to see, especially in Nothofagus pumilo forest, even in the understory, there's very little plants that can grow. And we think it's because of this nitrogen limitation. So basically, if you are an arbuscular mycorrhizal plant, there is no nitrogen available for you in the soil. So your only chance of surviving, I would say, would be to associate with these ectomycorrhizal fungi. And so the only plant that can do it there is Nothofagus pumilo. And so finally, as a take-home message, I hope that I convinced you that we found that actually Nothofagus in Patagonia, they can have very constructive, contrasting nitrogen economies in correlation with the availability of nitrogen and environmental conditions. So we were hoping to find a pattern similar to boreal forest, but we actually find it only in association with Nothofagus pumilo. But it's interesting because it's a pattern that was shown in the northern hemisphere in coniferous forest, while we found it here in the southern hemisphere with an angiosperm that is deciduous. And so before I take your question, I think we have time, I'd like to say to thank my sources of funding, the national agencies of the USA, of Argentina, and also from Switzerland, which gave me a postdoc fellowship. And also all my collaborators and especially all the students that brave the harsh climates to go do some soil sampling with me in Patagonia. And I'm happy to take your question.
That was wonderful, thank you. And with this weird situation where we can't clap, so I'll just clap on behalf of everyone. Yeah, so just if you want to ask a question, we've got that little Q&A box. You can put something in there. If you don't want to bother typing, just send a message into the chat and I can just unmute you or allow you to talk. Yeah, it was interesting, and I like the introduction slides as well, kind of explaining the background of it, and I always love a good mushroom photo. I just while I'm waiting for any questions to come in. So I did my PhD on rhizobia, these nitrogen fixing bacteria. So I was kind of thinking about that. So you hinted there that you did measure the microbial biomass of these systems, and hinted that in those waterlogged ones you had anoxic bacteria. Is there any role in these systems for as part of the whole nitrogen cycle, free living nitrogen fixing bacteria?
Yeah, that's a really good question, and that's actually something that I would really like to go back to look at this, or maybe because we have the soil sample, so we could look. Because we were really focusing. I was in the mycology lab, and so we were really focusing on the ectomycorrhizal fungi, and actually this pattern that we found in Nothofagus betuloides forest, we were really not expecting this. We were expecting the nitrogen, like the organic nitrogen economy, almost like everywhere, I would say. And so we never really focused on these bacteria. We almost no data on this. And so I tried to look in the literature. There is actually very little about this. But this is definitely something that I think that, that would be really interesting to study. Like, I remember I tried to look for literature about anoxic bacteria that maybe do nitrogen fixing. I don't know, maybe you probably know more than I do about this. But this is definitely something that we never focus on the bacteria side, but I think that in this type of forest, that's probably something that is. And the pH is extremely low. So that's the only thing. It's like 3.5 of pH is extremely low. Yeah.
Wow, yeah. We do have a question. Any AM fungi among the non-ECM fungi?
Very little, but I have to say that there is a slight bias here because we use the ITIS1 metabarcode, which is really bad to detect EM fungi. So we had very, very little. I think that probably that might be a problem to detect patterns in the Nothofagus Antarctica forest, because the understory is grasslands, and so there's potentially quite a lot of species of fungi that interact with this thing. In the Nothofagus betuloides and Nothofagus primulaforest, I have to tell you that even in the understory, there is almost no other plants present. There's a few mosses, but it's very few, like some seedlings. It's very, I know it sounds surprising, but it's really like monodominance in like the highest possible way. So I don't think that it's necessarily, they necessarily have a large role in Pumilo and Betuloides, but potentially yes, in Nothofagus and Antarctica forest, we should definitely try to probably use another loci to try to detect this, because the one we used, we were really focusing on the ECM fungi, I guess, and so it's like you say, you open the Panda box. Now we have all these results, and it opens to all these questions looking at our groups. But at the beginning, we wanted to focus on the ECM fungi. That's why we use these techniques.
We've got a question from Fabian, coming from more a comment really, but interesting. It's very interesting work. It's quite funny to see how Cortinarius genus seems to dominate Nothofagus dominant forests in different regions. And that's certainly the case in New Zealand as well. You know, if you want to see a court, you know, it's the beech forest. Do you have any idea why that might be? What makes that interaction so special?
Yeah, that's a good question. Actually, so I feel I have not had the chance to go to visit Nothofagus first in New Zealand. I hope soon. But what I've seen, for example, when I was in Tasmania, or some small fragments that we have in Victoria, is that Cortinarius is diverse and abundant, but it's mixed with, like, there's a lot of other things. They're like Amanitas, they're Versulas, they, you know, like other groups. While in Patagonia, this is really the one group that is highly diverse. To give you an example, there is, I think, 5 species of Versula in Patagonia. There is maybe, less than 10 species of Amanita. There's Ostropactylus, is the only bolete that is there. So it's really like there's only one group. And I feel that maybe there's something that has to do with maybe like some kind of biogeographic bottlenecks that happened, because the forests in Patagonia, they have been isolated from any other ECM plant from a really long time. So there's no, it's not like in Australasia where you have this all the Myrtaceae coming and some kind of connection with ECM forest, maybe in Southeast Asia, and like more like a tropical origin. So you have more mixture. In southern Patagonia, basically, when after southern Gondwana, the Nothofagus forest, they are there and they're totally isolated. So in terms of plant diversity in this forest, there's also a higher level of endemisms, for example. So maybe for some reason, Cortinarius was already abundant there and just stayed and get so diverse, and there is no other input from like other group. Yeah, I mean, that may be one explanation, but it's really strange, because it's really the only one group that is so diverse. In maybe some of the Ascomycetes, actually, it's not a lot of diversity, but some of the groups they have. It's a few genera, but they tend to have diverged also recently, like quite a lot. But yeah, I mean, almost no Amanita, Versula, like the typical Ectomycorrhizal things that you find. There's nothing there in this first.
Yeah, well, more chances to come to New Zealand and other places to study, I hope. We've got a question from Vanessa, really interesting, thank you. So if we find an area that has lots of Cortinarius species, is it likely that the soil is nitrogen poor?
Yeah, that's actually a really good question, because that's typically the one genus that have been studied to have these enzymes to access organic nitrogen matter, like they call peroxidases enzymes. It's not the only one, but I guess it's the one. I'm not sure if it's the one that I have the most studied, but it's interesting. I would tend to say yes until now. But then I think we also do. I mean, it's always difficult to make a generalization, I feel, because there's lots of species of Cortinarius. as we saw. And I mean, the different species have different requirements, right? So some of them may be really good at this, but some other species may be less. So I think when we look at such a huge genus, it would be really hard to, you know, to make generalities that all Cortinarius species are good for like are growing on nitrogen poor soil. I would say that definitely if the soil nitrogen is poor, the likelihood of finding Cortinarius is really high. But if you find a Cortinarius species, it doesn't necessarily mean that it grows on nitrogen poor soil. Yeah.
Another question from Eric. What are the mechanisms the tree uses to rid itself of ECM fungi if it has the nutrients and doesn't want to trade? Yeah.
Yeah. Okay. So yeah, that's an interesting question. And I have to say that physiologically, I don't really know what the tree does to get rid of the ECM fungi. Basically, they study, what they show is that if you give nutrient to a plant, like in an experiment, the colonization rate of the root is going to go down. But what is the mechanism that makes that the trees get rid of the mycorrhizal fungi? I'm not sure, because I mean, as we say, like, is it really a love affair, like I was telling, or is it really there's some point where we could also, I mean, I think the border was saying that the ectomycorrhizal fungi, it's actually parasiting the root or the mycorrhizal fungi, you know, and the some aspect, it's also something that there is. I mean, there's, of course, it's not a parasitic fungi because it's an obligate symbiont and it receives something from the plant and gives something back. But where sometimes the balance is a bit, you know, it's a bit fuzzy. So, but basically, yeah, it has been shown that if you give a lot of nitrogen to the plant, the colonization rate of the mycorrhizal fungi is going to go down. So there is probably potentially maybe some mechanisms. I would say that maybe if the plant doesn't give carbon anymore to the mycorrhizal fungi, it may end up dying as well. So it can be simply something like this. And also what has been shown is that if there is a lot, so if there's a lot of nitrogen available in the soil, then the arbuscular mycorrhizal plants, they also tend to thrive more. And so there's competition with the ectomycorrhizal tree. And that's why the ectomycorrhizal tree are going to be lower in dominance in places where you have more nitrogen available.
There's interesting thing that was done in the rhizobia world a decade or so ago on this very thing, and legumes can turn off oxygen delivery to individual nodules that are not supplying nitrogen. So, yeah, a huge amount of work would be needed. But yeah, there are those kind of fine level.
Oh yeah, definitely. Yeah. So it has been shown that if you, for example, in experiment, if you have, I think it was done with pine. If you have a pine and then you have some of the roots that, you know, they can separate them and then they inoculate with different species of ectomycorrhizal fungi, and they can really show that the plant is really directing. So if we receive more nitrogen from one of them, it actually gives more nitrogen. It's not it gives nitrogen to all the mycorrhizal fungi that are around. It can really say, okay, so I'm going to give you more because you're giving me more, and then the other one receive less. So it's very, yeah, it can be actually quite specific at the root level of when you see about like the trading of the nutrient in the roots.
Yeah, genius. Yeah, we have just a couple of minutes and people need to move on other things. Fabian has a question broken into different parts. I'm not sure if this is Fabian. I'm not sure if I well understood. Did you find some host preference pattern between your three species?
Yeah, so this is a question that I've been discussing this quite a lot with the researcher in New Zealand, because they were telling me that in some of the study they have found this host pattern. We didn't, but as I was telling in the talk, we didn't really study the specificity at fine scale because we were studying fungi in the soil. So we assume that if you find a fungi in the soil at the bottom of a tree species, we assume that it was associating with these tree species. But I think in order to really decipher if there's specificity, we should study the fungi that are inside the roots and not the ones that are in the soil. And also these three species, they belong to the same subgenus, or so the subgenus Nothofagus, while in New Zealand, you have all the different subgenus that are maybe more distantly related. So that might be something that explains that, yeah, the pattern in New Zealand were more like specific to host, but we didn't find any evidence, because in the mixed forest, basically, we didn't find differences between fungi associating with one of the species or the other species in the mixed forest. But it definitely needs to have more, yeah. And I have to say that in terms of specificity, I usually like to talk more about preference than specificity, because it has been shown that if you have a species, I have a colleague, actually, he did very interesting experiment in Israel. So in Israel, he's working with native pine and exotic eucalypts. And actually, if you give ectomycorrhizal fungi that is typically associated with these eucalypts to a pine in an experiment, and the pine has nothing else, I think it was a Rhedelomyces. So it's a Tuberaceae that is exclusively associated, like a southern hemisphere fungi. It associates with the pine, because if the pine has nothing else, you just take what is there, you know. So it's not the specificity. I think it's more like an ecological specificity than necessarily that there is a physiological barrier for the symbiosis. So, but yeah, no, we didn't find this in this first.
Awesome. Okay, I think we're gonna have to wrap up there as it gets into the hour. So yeah, thanks. Just a fantastic talk. Learn heaps. We have these talks every month, so yeah, just sign up for the mailing list and all of that. And oh yes, there's a question come through from me. Yes, if you are an AMS member, the recordings for all of these talks will be available to watch later. Thanks, everybody.
Okay, thank you.